Cancer, an unfair game? Surgery is the first choice in cancer treatment. For unresectable but radiosensitive tumors, irradiation is another frequently applied therapeutic option. Both methods have been improved significantly in recent years, including limb salvation and heavy ion therapy, to name a few advances. But metastatic cancer is a systemic disease and it cannot be cured using these local modalities alone. Surgery or radiotherapy is most often combined with chemotherapy in order to eliminate all cancer cells in the body and hopefully prevent the relapse of the cancer as well. It is truly a great scientific achievement that certain cancer types can be cured by chemotherapy. On the other hand, most metastatic solid cancers are still incurable by this treatment due to the acquired multiple drug resistance. Serious side effects and long-term toxicity are major drawbacks of this approach. Other anti-cancer treatments include hormonal therapy, immunotherapy, and molecular targeted therapy. These methods can be alternatively applied in simpler cancer types, but sadly, for patients with late-stage solid cancers, these special therapies have not proven to be curative either. Obviously, there is an urgent need for a new anti-cancer strategy. We have to look at cancer in a different way. Let's take a popular example, football. Here are the players and the substitute players of both teams. Normally, this is how a football match begins. But for now, let's suppose that the rules of this game have been changed unjustly. We have less, and the opponent has more players on the field, and they are allowed to have additional advantages as well. This is clearly an unfair game, as is cancer. To win the game, it is essential for us to know our opponent in detail, his major strength, his eventual weaknesses, his usual tactics and strategy. Based on the fact that we cannot defeat our opponent alone, we turn to some suitable external help and adjust our strategy. We realize that concentrating only on attack would be a major failure, so we organize our team to be efficient both in defense and offense. But how could a similar strategy work in cancer? In our first video, we discussed the three major pillars of carcinogenesis. Advanced cancer has already passed through these steps and is represented by large intratumor heterogeneity and circulating tumor cells. The presence of metastasis bears witness to successful neoplastic invasion and migration. Its increased nutrient flux is measurable and its growth is observable. So, what should we do about it? How could we defeat metastatic cancer? We have to adjust our strategy. Of course, it's easier said than done, and we have to consider following questions carefully. Which therapeutic concepts will form the basis of our strategy? How can we really stop tumor progression and eventually reverse or eliminate malignant transformations and return to normalcy? What tools, drugs, phytochemicals, treatment methods are available for us to use, and how can they be combined and applied most efficiently? Our new adjusted anti-cancer strategy would be based on the following principles and paradigm changes. Don't just treat cancer, tend to the soul as well, to keep a strong psyche and a great fighting spirit. A safe and tolerable treatment will not only be greatly appreciated, but may contribute to the success of the therapy as well. Instead of a rigid, one-size-fits-all treatment protocol, devise a unique, individualized, patient-specific plan and monitor disease status closely to be able to adjust the therapy quickly if necessary. Apply low-dose orthogonal metronomic therapy with low toxicity instead of using maximal tolerated dose chemotherapy with high toxicity. Don't try to kill all cancer cells obsessively, but also aim for control, because at this late stage there is an extremely small chance to achieve total elimination. And last, but definitely not least, increase the therapeutic window, that is, the tumor specificity of the treatment. This aspect is actually so important that we are going to discuss it in more detail. At a very low dose, drugs have practically no effects at all. With increasing drug concentration, the therapeutic effect increases as well, but above a certain threshold, side effects and toxicity start to intensify, thus inhibiting further increase of the dosage. Metronomic therapy operates mainly in this range, whereas chemotherapy in this range. Both approaches have their own disadvantage. This may be not effective enough, while this may be too toxic. We favor the use of low-dose drug combinations, so to really have a chance against cancer, it is absolutely essential for us to widen this therapeutic window. So, what are in principle the main options to achieve that? Different tumors have different vulnerabilities, so first the identification of suitable targets for treatment is needed. Molecular imaging techniques and an integrated genomic analysis of biopsy samples could be very helpful in this process. Furthermore, measuring validated serum tumor biomarkers and determining drug resistance of the tumor can supply valuable information to the development of personalized treatment strategy. Once promising intracellular or microenvironmental targets are identified, an intensive search should be carried out to find efficient synergistic drug combinations. The synergistic effect enhances treatment efficacy or permits the decrease of drug concentrations and thus reduces side effects and toxicity. Drugs selected this way should be optimized for proper concentrations and dosing frequency. As a rule of thumb, start at lower doses and up-titrate slowly. Whenever possible, avoid high-dose bolus administration and apply drugs continuously or as frequently as expedient. Each patient should get detailed dietary instructions taking into account possible food-drug interactions. This is especially important after oral administration, as drug bioavailability may vary largely depending on diet. The therapeutic window can also be broadened by supplementing drugs with functional foods or phytochemicals, which reduce normal tissue damage of the anticancer drug and may even increase cytotoxicity against tumor cells. In recent years, the concepts of oncogene addiction and synthetic lethality have been increasingly discussed in anti-cancer publications, and they are tested in different experiments. Proliferative, metabolic, and anti-apoptotic pathways are usually upregulated in malignant lesions, which enable rapid tumor growth, but also render these cells more addicted on protein signaling in these mechanisms, thus providing opportunities for selective targeting. In summary, we emphasize cytotoxicity should be restricted as much as possible only to malignant cells. We should search for drugs with larger therapeutic window and apply subtoxic doses. More than 100,000 drugs have already been tested against cancer cell lines worldwide. Many of these have been further analyzed and categorized based on their biochemical properties and mode of action. Unfortunately, successful laboratory experiments seldom translate to drug approval and even more rarely to definitive cures in the hospital. Nevertheless, it is not impossible to find efficient anti-cancer drug combinations and make plausible therapeutical suggestions based on those principles discussed so far. To illustrate our new approach, let us look at an especially aggressive bone tumor, osteosarcoma. Its pathogenesis and etiology is not completely understood. Remarkably, several studies have demonstrated that the simian virus may contribute to tumor development in a large number of patients. SV40 is an oncogenic polyomavirus found in rhesus monkey kidney cells and may have infected children all around the world in polio vaccination programs. Osteosarcoma patients with inoperable tumor or lung metastasis have a poor prognosis. Current conventional chemotherapeutic treatments with high-dose methotrexate, cisplatin, doxorubicin, ifosfamide, and etoposide rarely achieve long-term survival. Osteosarcoma is one of the most genomically unstable cancer types with frequent hypertriploidy. Because of its brutally complex karyotype, as you can see in the circus plots, it is extremely difficult to identify the main driver mutations. Such an extremely heterogeneous, highly malignant disease obviously requires a uniquely devised and thoroughly elaborated therapeutic plan. To check if we can come up with well-founded suggestions which could be implemented in the clinic based on our principles discussed so far, we scrutinized recent clinical trials in osteosarcoma on the NCI website and did an extensive literature search on google.com and PubMed using the following keywords and diverse combinations of these, as well as some other pertaining keywords. We found a vast number of scientific publications, 2 of which we mention now. In their excellent review article, Gasper and co-workers discussed more than 40 promising drugs against osteosarcoma. Valuable data can be found in the paper of Van Maldegem and co-workers about clinical trials in osteosarcoma and Ewing sarcoma. After an in-depth analysis of these and many other enlightening writings on the subject, we've prepared a map of possibly effective drugs and anti-cancer agents grouped by their types and targets. So, remembering our football game analogy, we have selected a small group of agents which could give better odds for patients to survive metastatic osteosarcoma. We can't discuss these drugs in detail here, but we can mention some interesting aspects and refer those of you who would like more information to browse the references listed at the end of this video. Metastatic tumor cells are characterized by softness and enhanced cytoskeletal plasticity. Paclitaxel is a plant-derived antimitotic agent which hampers cell motility and angiogenesis at low concentrations. According to recent studies, it also increases the intracellular levels of reactive oxygen species in cancer cells. A current paper reported that paclitaxel enhanced apoptosis of osteosarcoma cells in hyperthermal therapy. The albumin-bound form of paclitaxel demonstrated a high antitumor effect in osteosarcoma xenografts. Synergistic effects have been observed between paclitaxel and histone deacetylase inhibitors. Co-treatment of panobinostat with the autophagy inhibitor chloroquine resulted in accumulated tumor cell death. In vivo studies confirmed that HDAC inhibitors induce differentiation of chondrosarcoma cells. A 2013 publication reported terminal differentiation of osteosarcoma cells upon sustained low-dose treatment with panobinostat. A combination of HDAC and mammalian target of rapamycin inhibitors exhibited in vitro synergism in osteosarcoma and other tumor types. Everolimus, an orally available mTOR inhibitor, synergized with zoledronic acid to decrease RAS levels and reduce tumor development in 2 murine models of osteoblastic or osteolytic osteosarcoma. The everolimus-sorafenib combination yielded impaired tumor growth and reduced migratory and metastatic potential in osteosarcoma preclinical models. Blockade of the mTOR pathway with rapamycin or its analog led to a significant inhibition of ESRIM-mediated lung metastasis in a murine model of osteosarcoma. As we emphasized in our first video, metabolic flexibility is essential for tumor growth and invasion. Simultaneous inhibition of the mTOR pathway and glycolysis can synergistically reduce survival of cancer cells. Non-cytotoxic concentrations of 2-deoxy-D-glucose dose-dependently inhibited in vitro migration and invasion in a highly metastatic osteosarcoma cell line. It increased the efficacy of paclitaxel in osteosarcoma in vivo. Combined with metformin, it significantly reduced cell proliferation in high-grade glioma. Metformin is an antidiabetic drug, but recently it has been often employed as an anti-cancer agent. Metformin synergistically enhanced the activity of an HDAC inhibitor, trichostatin A, in osteosarcoma. Laboratory experiments indicate that metformin could actively prevent esrin-regulated metastasis formation in osteosarcoma. Adding rosiglitazone to metformin increases insulin sensitivity and reduces growth factor signaling. Rosiglitazone can induce differentiation in osteosarcoma cells. Another PPAR gamma agonist, troglitazone, upregulated PTEN expression and induced growth inhibition in osteosarcoma cell line. Rosiglitazone prevented osteoclast formation and bone resorption. Bone resorption is profoundly affected by the microenvironmental pH. The disruptive action of osteolytic agents like parathyroid hormone or prostaglandin can largely be counterbalanced by the addition of an alkali buffer. Sodium bicarbonate has been shown to effectively reduce acid-mediated tumor invasion. The transmembrane pH reversal is a crucial process in carcinogenesis. Therefore, clinical trials with other pH modulators are suggested. The inhibition of heat shock proteins sensitizes many types of cancer cells to apoptosis. The HSP90 inhibitor tenespamycin has demonstrated strong antitumor activity in osteosarcoma cell lines involving mitochondrial membrane depolarization and depletion of glutathione. Although it did not show enough responses in a high-dose clinical trial, at lower doses and in combination with paclitaxel or a WEE1 inhibitor in thermothermia, it could be quite efficient against osteosarcoma. The nitrogen-containing biphosphonate, zoledronic acid, has been shown to be effective in the treatment of osteoporosis, hypercalcemia, and metastatic bone tumors. Recent investigations have shown that it inhibits primary bone tumor growth as well. Zoledronic acid reduced osteoclast formation and maintained pain control in chondrosarcoma. Zoledronic acid acted synergistically in vitro when administered concurrently with paclitaxel or gemcitabine, not only in wild-type osteosarcoma cells, but also in P-glycoprotein overexpressed drug-resistant osteosarcoma cells. Gemcitabine is a nucleoside analog DNA synthesis inhibitor. Its targeted delivery to the lungs inhibited the growth of osteosarcoma metastases in mice. Aerosol gemcitabine may benefit osteosarcoma patients with pulmonary metastases. Gemcitabine showed enhanced efficacy in combination with several anticancer agents. Inflammation fosters cancerous evolution, and the administration of nonsteroidal anti-inflammatory drugs like acetylsalicylic acid can mediate this effect. Recently, it has been shown that aspirin, dose-dependently, inhibited cell growth of human osteosarcoma line. In another experiment, aspirin, together with an HDAC inhibitor, synergistically killed hepatocellular cancer cells. It must be emphasized that this combination of drugs has never been tested, and its implementation should be carefully refined, depending on a number of parameters not detailed in this video. Nevertheless, we believe that patients with inoperable metastatic osteosarcoma could significantly benefit from an individually tailored, low-dose orthogonal therapy based on our suggestions. Such a treatment should be feasible as a pilot study or a single-person trial with good tolerability and low toxicity. Its efficacy could eventually be further increased by augmenting or partially replacing it with cyclotherapy, chronotherapy, or immunotherapy. In the future, immunotherapy could even be by itself the solution for cancer treatment, but not yet. We urge you to read this article by Daniel Angber, which is not only illuminating and very well written, but has far-reaching implications as well. It is a story about Rolf Steinman, who was diagnosed with pancreatic cancer in 2007. As an immunologist, he knew exactly that existing treatment protocols had no curative potential for his disease, so he decided to try new therapies. He had his tumor's genome analyzed for special vulnerabilities. He initiated cell culture and mouse experiments for drug resistance in a number of laboratories. After careful evaluation of the data, he designed an immune-based therapy for himself. He applied for and got approval from the FDA for his one-man clinical trials. Over 4 years, he overtook as many as 8 previously untested therapies. Steinman was posthumously awarded for the Nobel Prize in 2011. We still don't know which strategy is the best against advanced solid cancers, but the cooperation of innovative and dedicated physicians and researchers could bring us closer to the solution. Express your critics and suggestions. Cancer patients need your help now, because the question remains, how could we really cure cancer? Let us think together.